Oh, okay, let's start. Anyway, at the beginning, I will just make a recap, so I think it's better to start. So, good morning. Today we'll continue to discuss about programmability in software matter. And today I would like to bring you a bit to the world of nanotechnology. And I will show you a few other examples of all these ideas. So just to recap, we saw already, already my two dogs running on fish. You can see that the interesting software matter with interesting properties. And yes, living systems are soft matter because they are made of soft matter components, but also because biological process work with the thermal energy level. So, as I told you, so what is interesting with software matter is we can have some kind of global view of all these systems, whatever universe they are, and we can have some kind of laws that can describe software matter, even though the systems can be so different. So this could be quite interesting if we can use it to approach living systems. And as I said in this course, and trying to not to drive, not to do biophysics, all these kind of things, but more interesting are not applied properties. So dynamic self-organization, this is something I will talk to you about. Sensing, reconfigurability, something that I will discuss a lot. Adaptability, motion, this is something we will explain. And we'll use only synthetic systems. It's something that we control very well. And we have two objectives. One is to understand how systems are working, and another one is to make. To know which we could build synthetic matter that could require such lifelike properties. Today we'll be a lot in making. Okay, we'll not be so much in a bit of understanding and a lot of making. And as you know, functions ensure by protein. We saw many things yesterday, and it's programmed by DNA. And this is how life is organized with a simple molecule that is encoding information. Interesting is that you have this separation of a program in a simple molecule to DNA and functions in the form of proteins that are so diverse. And we saw that it's interesting that both this DNA and proteins are software matter objects. So we can use these properties to manipulate them, and it's also something we do today. But they are also subjected to soft interactions. And this may be quite interesting because they can have some reconfigurability and dynamic, for instance, conformation. Something that will be also quite important for today. And with that, I say it's quite interesting now because we have this knowledge of soft matter. We know that living systems are part of it, and we are interested in properties. So, and then we can raise this question or this challenge: can we build these such lifelike properties, such as reconfigurability and so on and so forth, in synthetic material? And instead of doing formulation, as I explained in today, we will bring a program so that we can control the property of the synthetic materials. And I think we can use DNA for two things. We can use DNA to program functions. This is what we saw yesterday. But we can also use DNA to program structure, and this is what we will see today. So this we saw. We have now quite interesting object that is synthetic DNA that is highly accessible, that you can synthesize by yourself, or you can just order from a company, and it's actually inexpensive. So there is immediate accessibility of DNA, and what is really nice is that it's fully programmable. You can get any sequence you want. Just need to tell your provider, I want the ACC, AT, GGC, AT. That's it. So you have this programmability, and also you can add on DNA additional functions so that you can make this break functional. So you have this combination of accessibility and programmability that makes DNA very interesting and very synthetic DNA interesting and very special. If you just arrive, don't worry, I mainly made a recap of what we have seen yesterday. So now we'll start to see a little bit of new things. So we saw that we could use synthetic DNA as a code to program functions or structures. Functions will give rise to synthetic biology, it's a bit what we saw yesterday. And structures can bring to these methods of structural DNA nanotechnology, and this is what we will see today. So, as we saw yesterday, we have this path to use DNA to program a function. And what if we think about what kind of program we use? We saw that in fact, we don't use so much this aspect. We use the fact that DNA is in support of a code, of a genetic code. That's why I call it genetically encoded matter. So in nature, natural DNA is supported with genetic code, and in synthetic biology, we reuse synthetic DNA to program sequence-based gene writing. Yeah? Yesterday we were writing sequences of proteins with DNA. So yesterday, if I wanted to do a given protein, I have an enzyme that's interesting, I get the sequence, and then I take the DNA, and my DNA will be ATT, ATCG, ATT, ATCG, and this will be my language. This language will be transformed into a protein. So we were using DNA as a sequence-based language to write genes, to write sequences that will be the description of my protein. Okay. But we didn't use at all the structure of DNA, and this is what we use today. DNA is also a simple structure, but it's quite interesting because DNA works by specific base pairing recognition. So in nature, we have double helix that is organized through hydrogen bonds. Through this specific recognition, A bound to T through two hydrogen bonds, G binds to C to three hydrogen bonds, and A cannot bind to G nor C, T cannot bind neither to G or C, G cannot bind to adenine or thymine, and C cannot bind to adenine or thymine. So you have this very specific recognition. So you have 4 breaks, but they can bind only 2 by 2. Okay? This will be our way to writing, and this will be our sequence reprogrammability. So in DNA nanotechnology, we use synthetic DNA, but in this case of programming, will not be by writing a language that will be a sequence for protein, but it will be to use it to do sequence-based recognition. And this is what we will do in this. So what do I mean by sequence-based recognition? Who wants to come? Yours? Yes. Come. So, can you explain the sequence-based recognition? Yeah? No? So, we say that A recognize T, right? And C recognize G. Okay? So, can you draw DNA? Oh, yeah, this is big. Okay. Yeah. Yeah. Yeah, it's not, yeah, it can be a bit more round up, yes. G, A, okay. Okay. Yes. Okay. And then. Yeah. Yeah. And. Yeah. Okay. So it's T A C. Okay. So okay. So yeah. Okay. So for yeah, it's good. So it's very important. Yes. Thank you. But for practice, it would be a bit more simple. We will write like that. Okay. I will write like that. It will be a bit easier. So I told you I can buy a DNA oligo to wherever I want. So I can buy, for example, this sequence. Okay. So could someone come? No, I would like to buy a sequence that will be self-complementary. I want to buy a sequence that can bind to itself, so can somebody can to make it? For example, yes, 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 you can come. Okay, so we'll start with this. So I want a sequence that is self-complementary. Yes. Yeah, because this doesn't work. I want to. Alors, you can make a strong, and yeah, we can start like this. Yes. Yeah. So here I have two strands. Okay. So now I want self-complementary strand. It means I want to buy one. Okay, but they will also buy two by two. Here you have complementary between two DNA, okay? Yes. Yes, exactly. Okay. Yeah, who wants to try? You? Okay. So, no, yeah, I have a quick small question for all. So, you see, I take this DNA, right? And then let me turn it like that. Okay? So you put A, G, C, C, A, A, G. So you agree that it's just it's the same, right? So my question is this guy, will it bind to this guy? Yes? Who say that? Who say yes? Who say yes? No one? Everyone say no. Who say no? Okay. Ah, there are great people. So you say no. So everyone, because you know, maybe you can bet something. Okay. So that would be nice. So I said I take this sequence. This sequence is just I can just make like this, right? It's the same sequence. So my question is, this guy does it make double helix here or not? No? Yes? No? I have difficulty because in India you do this to say yes, and in France you do this to say no, so I'm a bit confused when I say, well, I don't know if it's yes or no. So the question is very simple. I ask you, you can be wrong, or you don't know, maybe you don't know. Okay. In fact, the answer is no, because something that was difficult and to draw is that actually DNA is a chiral molecule. And when DNA is polymerized, you have directionality. So in DNA, you have a direction. So that's why the DNA is how it is polymerized, okay? So if you want to make a double helix, this guy should be like that. Okay? So it means that when I will order this sequence that is complementary, I will send as a sequence to the provider this guy actually. Okay? AGCAAT. So in fact, if I want to be complementary to this guy, I will order this sequence and not this one. Okay? So that's why, to be convenient, sometimes I will make an arrow like that. Okay? So it will be the direction from 5' to 3'. Okay, you can go back, okay? So in this case, if I order this guy, it means that if I want the complementary molecule, I will buy A, G, C, C, A, and T. Okay? So this is how I can make two complementary strands. So just my small question at the beginning was, can we have a self-complementary strand? So an oligo that will be complementary to itself. So can someone want to draw? Maybe somebody else? You want? You know, when you sit here, there is a high chance that, yeah, you come. So, you don't understand my question? Okay, but then you will know. So, this guy is complementary to this guy. Okay, so this plus this will do this and this. Okay, with A, T, T, G, G, T, A, C, C, A, T. Okay. So now I would like to take just one molecule with the sequence of your choice. I want this guy to assemble with itself. Okay? So I don't want to use two, but just one molecule. Can you imagine the sequence? Do you understand the question? You don't understand the question? Okay, I'll be trying to explain. Here, I have this. Do you understand this? Okay. So I take this, and I take this, and the self-assembly. Now I want to take this, and the same oligo, I want it to be complementary to itself. It's called self-complementary. Okay? So for example, if I have an A here, if I have an A here, if I have an A here, draw the complementary sequence. This guy. T, but why this is complementary to here? No, I want this guy to be self-complementary. Okay, if it's too difficult, I will try to take this over X. So, you want to try? Huh? No? Even if I ask kindly? No? Okay, no problem. So you want to come? No? Whatever. Nobody likes DNA, but DNA, you need to like DNA with me. It's, yeah, welcome. We'll do together. Okay, we'll do together. It's not. It looks simple. It's not so. It's not so simple. It's just a question of understanding a bit the language of DNA. So that's why I'm doing this, because after I will show you many things. So, okay. So do you understand the question? This molecule is an oligonucleotide. I want this molecule to assemble with itself. Okay? Okay? So, if I have an A here, okay, can you draw the complementary strand? Draw it here. Yes. Ah, but that's very good. Yes. That's very nice. So if I have A here, what should I get here? Here. Yes, the T, yeah. T. Don't be shy, write the big T. Yes. So, yes. Wait. Look. Isn't T. So. No, look at it. This one is here, and this one is. Yes. Do you see? Then it's nice molecule. Okay, so you can continue. You can make any sequence you want. Don't go too fast because there will be mistake, huh? Okay, so if you have T, don't go too far because it. Yes. So here you have. Yes. So maybe put it here because otherwise it will be finished. Do you understand? The second is this one. You see, this is the head. You should write the vowel bigger. Okay. So if here you have an A, this should be. Okay. So here you have the C. Okay. Yeah. No. Look, it's close to the head. Okay. T, A. And here. C. Yeah, and C. Okay, and here you have A, T. You are at the back, right? You are here. So you have A, T. You see, it works. And then you have G, T. So you see, you are taking this sequence and they are complementary. So you just have two left. So go for whatever you want. Whatever. Yes, C, yes. So the C is here. Well done. Bravo. The C. So it looks, at the beginning we see it's easy, just like it together, but you see that you have to remember there is directionality in DNA. It's very important. And then also double helix. So you see that if I order the sequence, it will, if I mix, they will self-assemble with self, or it can be self-assembled, or they can assemble together. So then an important point I will just want to present to you before showing more elaborate things we can do with origami. Is so now you saw how DNA self-assembled, and these are controlled by hydrogen bonds. And hydrogen bonds with very soft interactions, so it's strong but not so strong. So with temperature, it's easy to break hydrogen bonds, and it's also easy to reassemble. So whatever DNA I will take, if I increase the temperature enough, at some point, hydrogen bonds will be broken, and I will have two strands separating. Okay, and this is called melting. So here the DNA is hybridized, and here it's melting. Okay, and this is a quite cooperative process. So this will be defined. So if you measure, for example, you measure, for example, something that represents behavior, we have something like that. This is temperature, and here you have double helix, and here you have two separated bonds, and this will be your melting temperature. This will be the temperature at which your double helix will separate into two strands. It's a very important point. So now, this is due, you need energy to separate these two strands. So now I can ask you the question, what happens if now I increase, or I want to measure, for example, the melting temperature of any double-stranded DNA, and this is the length of DNA. So I increase the length of DNA. What do you think will happen to the melting temperature? So if I have a DNA that is longer, will it be easier or more complicated to separate these two strands? Harder? Yes, it will be harder because it's like a zipper. Okay, if you have a long zipper, you will need to. So it's a bit like a zipper, but it's between a zipper and a sticker, if you want. So it's a zipper in the sense that it's cooperative. When you start, it will be easier. So at some point, it will saturate because it's cooperative. But you have also all the adsorption bonds. If I increase the length, the melting temperature will increase, and at some point, will become constant. Okay, so because it's cooperative, at some point, when you start to open the DNA, everything will be open. So it depends on the length of DNA. It depends on the sequence. This is not so important, but in general, in AT you have 2 hydrogen bonds. In JC you have 3. So in general, the melting temperature of GC-rich DNA will be larger than the melting temperature of AT-rich. So now, if yes?
What is the y-axis of the first plot actually?
Here?
Yeah.
It's a measurement of something. So it could be, for example, you can use, it depends what you measure, you have techniques. For example, you use a fluorescent dye that goes, when it goes inside, it's more fluorescent. And then the fluorescence the dye will be removed, the fluorescence will decrease. So it could be a fluorescence of the dye. It could be absorbance. In this case, it can be the opposite. It absorbs a bit more like here, so in this case, it will be like that. It could be a threat, so you have 2 molecules here, so you have fluorescence. For example, if you have a threat pair with a donor and acceptor, here it will be fluorescent, it will be not fluorescent. So it's something that measures. There are many ways to measure it, but that's why I didn't write this. So if I increase the length of DNA, my melting temperature will be higher. If I have more GC, the melting temperature will be higher than by 80. And what about salt? If I put more salt, like any kind of salt in my solution, what do you think the melting temperature will do? Will it increase? Will it decrease? What could be the effect of salt on DNA? If I add salt on this molecule, will it stabilize or destabilize the molecule? Do you have any idea? Huh? I cannot hear, sorry.
Melting temperature decreases.
Melting temperature decreases. Why?
The melting temperature reactive temperature.
So you think the melting temperature increases because
Decreases.
decreases. So it means that if it decreases, it's easier to separate DNA. So it means that salt destabilizes the DNA.
Yes.
Okay. Who agrees with this explanation? Who disagrees? You are very shy today. You can have your opinion. DNA is a good electrolyte, so it's full of charges, it's full of negative charges. Okay? So if we are alive, all of us, it's because in each of our cells we have a lot of salt to screen this repulsion. So all the biopolymers are electrolytes, highly charged. Okay. So if this electrostatic interactions were not screened, we would be just exploded because they would repel each other. And you know, you have to assemble a negatively charged molecule with a negatively charged molecule. This is DNA. This is the two negatively charged molecules together. DNA has the highest charge density in nature, highest linear charge density in nature. You cannot find any cable with so much charges on a given length. So this is the highest charge density in nature. And you have to put two together. Okay, and there is no ions like in between to stick like this guy. So the only way to do is to screen the reactions or the balance. And that's why in nature you are at around 100, 200 millimolar. The balance is 1 nanometer. And what is the size of a double helix is 2 nanometer. Okay, so you are just at the good distance where you start to be not too annoyed by this electrostatic repulsion. So when I will increase salt, I will screen more the interaction. Okay? So will it stabilize the molecule or will it destabilize? It will stabilize the DNA, right? Because you will have less repulsion, electrostatic repulsion. So if it's stabilized, it means that you need more energy to melt the DNA. So it means that the melting temperature will decrease. So salt affects a lot the melting temperature, like pH, like other parameters. Okay, so we saw a bit of this principle of base pairing recognition, and now I will show you what we can do with it, and how we can make some objects out of it. And this will be now the second part of my talk. Okay, so I will show you different things, and we'll see a lot of DNA origamis, and I will just start to show you how by using this principle of DNA-based pairing recognition, we can build objects. And one way, there are many methods, the method I will show you a lot today, is called the DNA origami. This is how it works. You start from a single-stranded DNA, which has some kind of sequence, and then you will add a cocktail of molecules. I will show you once I will play it again. And this cocktail of molecules will program the folding of this initially disordered molecule that is circular DNA into a target shape. So here, for example, it's a rectangle. So I will just play it again. So here we are. We start from a single-stranded DNA. So you see it has no particular structure. It's circular. And then, so imagine I make a zoom. I have locally this kind of sequence. And then I will take a small oligonucleotide as we have designed. And this nucleotide has a sequence. Okay, so you see my oligo, I designed it so that it's complementary to this part. So My circular DNA has also direction. So for example, this could be the 3' end for the right arrow. So I will design the sequence compare, but only to this part. And this part, it will be complementary, but to another location of the circular DNA. So if I continue my video, so of course we have another one that is binding here, another one that is binding. So I'm looking at this oligo, and you see this oligo doesn't bind here, right? It will bind to another place. And it will bind far away. For example, here. Okay? So, because it's binding here, it will bring these two parts together. Okay? And we call it a staple, because it really works like a staple. It will bring two parts together. And then maybe the last part of this oligo will bind to another location far away, for example, here. Okay, so you see that if now I play it again, and we just look at this specific oligonucleotide. In this sequence, one part is recognizing this part, and another part of the oligo is recognizing another part here, and the end of this oligo is recognizing this part here. So this single oligo will bring these two parts together, like a staple, okay, like bring together. And I will not use one oligo, but you saw that I use a cocktail. I will use 300 of oligonucleotides, and they will all take all these parts of this scaffold, bring together, and make the shape I want. And this is called DNA origami. Okay, so you saw that in this method, we are using a circular stranded DNA. So I'd like to ask you whether you know why, what could be the interest of using this. So you see that here, I start something like that, and then I have sequence here, and this is A, this is B, and I will take an oligo that this part will be complementary to this part, and this part will be complementary to this part. I put a bar on it's complementary, and then it will bridge. Okay, so it will make something like that. Okay, so it's a statement. And then I will use many of them so that at the end I have the shape I want. For example, a rectangle with all my staples inside. Okay, so this is how it works. So my question is, do you know why we just don't do like, I don't know, something like that. Why could be the interest of using this circular DNA and not just oligos together? It's not an easy question. I can tell you that the idea of this nanotechnology started by a guy from Leipzig in New York in the 1980s. He said we could build organized matter with DNA. It was really amazing. He wrote papers that could be great, but in fact, this guy could make for the first time a cube out of DNA. So he took some oligonucleotides that could make a cube. I did that in the 1990s, so much before I was. But it was very difficult to make complex objects. And one day, in 2006, a young guy in California say, a computer scientist actually, say, if we take this, it will work much better, it will be much easier. So why is it easier to use this scaffold, because the scaffold is technology, than just to use some oligo that are assembled together? Can you imagine what would it be easier to do like that compared to just assemble oligos? Why could it be difficult to do that? Why cannot just mix? Here I have 300 oligonucleotides to make an origami. So why I could not just make the oligo together, 300 oligonucleotides, all different kind of scaffold?
Oligo, I can take it one layer, but how do I take two complementary oligo types and I put them together and they form. It forms in one, the two layers of two lines on the top. How do we get the third layer assembled to it?
Yeah, but you could imagine like what I draw, that this guy could be like that. But then I have a double helix, okay? So what people, to make this kind of thing, they have the idea to make crossover. So when you do crossover, it means that you will take DNA that will do this kind of things. So it's possible to make, for example, there is a way to do only oligo. It's called tiles. So if I do like that, I don't know if you see, but you see if I do like that, I have possibility to make 2D object, right? You understand? So this is called a tile. It's called SST, single-stranded tile. So it's possible to build objects just with oligo, okay? So it's possible to do that. So you are just tiling. So it's true, if you just do this, you cannot do. But if you do like hairpin, you can make 2D. You can even make 3D if you make proper design. But this is also possible. I will show you an example. But this is much more difficult than this. And the reason is that you have to imagine that if you just list oligo, you need to have perfect stoichiometry. You need to have exactly all components at the same composition. And at the same time, you have to do this assembly without any mistake. But there is a high chance when you assemble your DNA that you have maybe a so small mismatch somewhere. So the oligo will bind to another one, maybe there is mistake, and then all the assembly is over. So with this kind of approach, it's very difficult to have good yield. So you will form objects, but maybe you will have a few percent of complete structure among many non-complete. And why it's really great to have the scaffold is that I will take for one molecule of scaffold, I can take 100 times excess of staples. And this is making it work so well. This is why you can have high yield of assembly. Because now, this will be controlled by this guy, and my excess of staple, I will have a huge excess of staple. So it means that this will help me a lot to correct mistakes, to have a good yield, and at the end, I will have almost 90%, 95%, nearly 100% yield in terms of this guy. All these guys will be perfectly formed. And if I have an excess of my staples, and let's say 100, maybe one staple will have an excess of 99, maybe one of 105, maybe 180. I don't care because there is so much excess. What is important is that they find all their targets. So what was the great breakthrough in DNA technology is when people have decided to use scaffold, and this is called DNA origami. It's a single Osler paper that makes this, and it was quite a revolution. So Now I'm going to show you a bit what we can do with these guys. Where I should put my remote controller. Okay, so now you have some idea a little bit how it works. So geoorigami technology is this single author paper 2006, and they show that you can make a lot of shapes, and then this field has exploded. So now there are so many people making things. And what is great is that it's a method that is working. You know, it's not just a method for a paper with like impressive paper. It's something that is actually working very well. So it's very easy to do DNA origami. You just order your DNA, and you can build any shape you want. Okay, so basically, you design a shape, then you draw your DNA, your circular DNA, then you position your staples, and then you order your staples, and then you can make your origami. So this is just the image from the first paper, but if you look on the internet, you will see people can do 2D, 3D, rabbits, Coca-Cola bottle, sphere, context objects, Mona Lisa face, and you have really amazing control of the morphology of your object. It can be DNA origami, it can be tiles, and I showed you can also make structures without scaffold, nanotubes, grids, and so on and so forth, but I will show many origami today because it's very robust method. And okay, it's very, when you are a scientist, it can be self-satisfactory to build this beautiful object, okay? You order and you build and have satisfaction. Like if you make a Lego brick, or if you make knitting, or if you're painting, you order your bricks, you assemble, you are satisfied at the end. But what is really interesting as a tool is that, in fact, as I told you yesterday, when I order an oligo, whatever the direction, I can ask the company to add a chemical function. For example, here, or here. This function can be a protein, it can be a particle, it can be a peptide, it can be anything you want. It can be a group to make click chemistry, it can be some azobenzene, it can be an amine group, it can be a carboxylic group, whatever you want. So you can, because now you know where is each oligo in your final structure, you can know, you can position objects wherever you want in your origami. So now you have a breadboard, you know, like electric circuits, where, or Lego if you want, and you can decide exactly where you will position your object. This could be particle, this could be protein, this could be function, and whatever else. It's really like a toy. And you can really position exactly as you want in 2D or in 3D. So imagine that there is no way to assemble objects in such controlled way as far as I know. You can take particles and you want to put 4 particles in space in a specific arrangement, and you want 2.4 nanometer between 2 particles and 7.3 to another one, and you want that arranged into an helix because you want to do some physical, for example, here people do some meta-molecules by magnetic particles, you can photonics, you can put protein together to study cascade reaction, you can study how protein interact, control their distance, you can make multi-ligands, you can put 1, 2, 3, 4 proteins, protein A, B, C, 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 A. So it's really a tool where you can organize matter in a very programmable way. So this is something very interesting as a tool to build objects. So the thing is, if we consider this from a soft matter point of view and the point of view of self-assembly, There is a small problem, and this problem lies really in how these objects are assembled. So if I take all my DNA molecules that I order, so I order on the internet, my scaffold, I order on the internet all my DNA strands, I put them together, I will not get this origami. I will just get misassembled structure because there are too much possibilities. You have to imagine that in one origami, you have 20,000, 30,000 hydrogen bonds. So you have 20,000, it's exactly the same interaction, 20,000, but each of them has to be exactly as good position. Imagine the number of the complexity of this energetic landscape when you need to assemble. It's half-full. So the system by itself is not capable to find a good position. It will make too many mistakes. So you will have just some balls of misassembled objects. So what people do to make it work, and what they have done for 20 years until now, is that they use thermodynamics. So it means that you take your soup of DNA, you heat above the melting temperature, what I explained to you before, so you break all the hydrogen bonds. So you break all the possible mistakes. And then you will do a very slow cooling down. So you will cool down for hours, sometimes for days. And then all DNA will find a good partner, and then you will have the folding object. And then you will have beautiful waveform object. So this method works very, very well. Highly, very reliable, and so on and so forth. But then, if we put this in this context of lifelike assembly, there is a problem. First is temperature. You need to heat above the melting temperature of DNA. So now, if I want to assemble a DNA origami inside Dr. Subramanian, you don't know the I will have a problem, he will have a problem because I have to boil him. That's a problem, I cannot do in situ assembly. I cannot do this lifelike properties. Life doesn't work at 80 degrees. First, and second, life works at constant temperature, right? Body is constant temperature. So there is a problem here in this temperature condition. The second problem is the time. Like your method imposes you some time. So you have to heat, and then you have to do this very slow cooling down. So you cannot really grow structure like that. And finally, the subjects, so you heat, you cool down, but then they become very stable. So you have beautiful object, but then this object is just this object forever. So it's great, but then you have zero reconfigurability, zero. Okay? So in this idea of lifelike properties, we are very far from that. So this is a problem for us, but it's also a general problem. Like this self-assembly method is quite a constraint, and this is what all the labs in the world are doing actually. So I think it could be interesting to find a way to do all of this in a way that could be at a fixed temperature, and that could be very gentle. That could be done at 25 degrees or body temperature. Just something that's really self-assembly, like a micelle, but we want to do not a micelle, but not an origami. So I will show you some examples of what we have done. And I will probably maybe talk about these two things, just to save your time. Just a very small first approach, a bit bold, a bit lengthy approach. So we say, okay, let's start simply. Let's see if we can do this reconfigurability from origami that are already prepared. And if we see we can just fold this origami, so like make an origami from an origami somehow. And can we make it simple? And this we just use soft matter property. So the experiment is very simple. We prepare the origami, and we insert them on the surface that is cationic. And you see that if your origami insert on the surface, origami is negatively charged, but on the cationic surface, then it will just adsorb flat. And then what is very interesting is that if now you take here a soft cationic layer, so just polymeric layer, you make a multi-layer. This layer is soft, it has the same amount of charge, it's cationic also, you have the same amount of charge, but now when your origami will assemble, because the surface is tough, it will fold on itself spontaneously. So origami at a rectangle will become this little tube. So you have this spontaneous suprafolding of the origami on itself just by electrostatic and polymeric interaction. And then because it's soft matter, this is reversible. So now that my guy is folded, I can add a competitor. So I add, in this case, heparin. It's a negatively charged polymer. And this negatively charged polymer will compete with the layer and my origami will assemble. So you will see AFM images that are done in situ. So we are imaging the origami in situ. You have the time, and you see that when I am made my origami open. So this you can do with many shapes, and you can fold and fold. You can put protein, you can fold and recover the protein, and so on and so on. So you have this reversible origami folding, so you have kind of reconfigurability. Right? But we are not something that is very generic. So now I would like to have a method to really have origami DNA self-assembly that could work as a summary. So to beat this problem, you really need to have such kind of method. And our idea was to say what was the problem with DNA origami is that people put conditions to have stable origami. And what they do for that, they use a lot of magnesium, so equivalent cations, we got a lot of salt. So they have conditions where the origami at the end are very stable. And then they put a high temperature to break this origami and cool down to find again this very stable state. So the problem, as you see, is that if you design a system that is overstabilized, you need a lot of temperature to destabilize it and reform it. That's the first problem. The second problem is this guy will be very static. So what we say is that if we could think about just simple conditions where our system is stable enough to assemble, but has some reconfigurability so that it can correct mistakes, then maybe it's possible. And we say electrostatics probably is the best way to do it. So let's try to adjust ionic conditions and electrostatic to find a way where the DNA will be stable enough to assemble, but not too much so that we have reconfigurability. So we just use salt. The simplest salt we can imagine, kitchen salt, sodium chloride. And if you adjust the concentration of sodium chloride so that you screen the electrostatic repulsion so that DNA can come together, but you don't put too much so that DNA can correct the mistake in terms of base pairing, then the system can assemble at the same time, correct its mistake to try to find the best assembly possible. So, and this works very well. So if you just mix your DNA mixture, put sodium chloride, just put on the bench, and the origami will fold itself. You don't need to heat, you don't need to cover. That's quite cool. And you can, with the DNA programmability, you can do any shape you want. And there is very good yield. And you can do many other things. You can do 3D, scaffold-free structures like grids. You can write letters, not so important. And quite interesting, we wanted to know, okay, it's great. It's the first time we can have some assembly on DNA origami, but how can it work? So to do that, we wanted to see the DNA folding in real time. It's not so easy to see real-time assembly in nanoscale. So we use atomic force microscopy. Atomic force microscopy, what you do is you need to adsorb an object and then you scan, right? But the problem is that if your origami is adsorbed, then it cannot assemble anymore. So if you adsorb strongly, then your object will not continue to evolve. So to be able to image this, what we did is on the surface we put a bilayer, so a lipid layer that's very soft. So we pre-form the origami like that. We put cholesterol. So cholesterol will go to the bilayer. So now I can bind this part on the bilayer, and this part is free. And then at time equals 0, I add my staples, and then this guy can assemble below my IFF. And I can do it for real-time imaging at nanoscale level to see the folding of the linear. This is the first time that people could see folding of linear origami in live. And what is very interesting, what we see is that, in fact, if you look at this guy, you can see that it will fold from one corner to the other. If you look at this guy, you will see that it folds from the inside to the outside. If you look at the other guy, you will see that it folds from the outside and at the end will close. This is just an example, but it means that you have not one single folding pathway, but you have a large number of folding pathways. So what happens, actually, is that you have this very complicated energetic landscape. And by working at this soul concentration, I can flatten a bit this landscape, so my energy minima are not too deep. And so what happens is that my origami go a little bit lower in energy, and then it can always escape. Even if I have local minimum, it can always escape, go a little bit down, escape, go a little bit down, escape, a little bit down. And we find the minimum from energy that is here, for example, this is this shape. But maybe another assembly will go here, another local minimum, another kinetic trap. But this is not too deep, so it can escape. Maybe this guy will fall in a totally different way, but at the end, we always find the free energy minimum. And this is very nice, because then you just put this down in this very complex energetic landscape, and you can find by itself the energy minimum, and it can escape all this kinetic trap. So we have seen that it's actually a thermodynamic control. So, because it's thermodynamic control, you can play with it. So, for example, you can say, okay, if I take a scaffold and I put a program for triangles and a program of rectangles, what I will get? I can ask you, but you have no way to guess, so we didn't know even. And what we got is a single shape. Triangles only, and we see that the system will make only triangles. Okay, we put both, and they make a triangle. And what is very interesting is that if you look at the free energy minimum of these two shapes, this one is a little bit lower. So you have a system that by itself is capable to find what is the most stable conformation. But even though it's a very, very complex, imagine for one shape, 20,000 hydrogen bonds, a huge number of possibilities. Imagine when you mix two cocktails, how complex it is. Okay? Imagine you take two Lego blocks, okay, and you mix everything together, and you don't know, you don't have the map to know how to assemble it. You shake, okay, so you have one Lego mix that makes Star Wars vessels, and one Lego ship that makes a dinosaur. You mix them together, you shake, you open, and you just have the dinosaur. Okay? You saw how complex it is, and it can do by itself. So then we can say, okay, maybe they can evolve this object, because now they can select. So if we start from the stable, from a shape that is really stable, we add a competitive shape, and then you'll see that origami that transform. And it's the first time we can see origami that transform. You see, they are rectangle, we add the competitive stacker, and they transform into triangle. Okay, so you have really no possibility to have evolutive structure, and everything is at temporal and fixed temperature. So you can do things to make really nice things. So this is what we have as a method now. We have this isothermal assembly of highly multi-component compounds. You can make any programmable shape you want. You can incorporate proteins wherever you want, and you have the system that is capable of selecting the most stable shape and also capable to evolve. And I will show you now a few examples of things that can be done with this system in the last few minutes. I will show you some examples of all this live DNA assembly we can do with this isothermal assembly. First, we can just use this origami as dynamic scaffold. Okay, so I show you that they can transform, but you see in our first experiments, we were beginners, and it was very slow process. So it was isothermal, but takes 42 days, so a bit slow. So we try to improve and by playing on electrostatics and so on and so forth. Now we have conditions where we have very nice transformation in just a few hours. Origami transformation is something very difficult to do. It's actually the first time it's done, and now it can be done in 3 hours, and you can play with the programmability. We can have fun. You can, for example, transform a 2D shape into another one, like morphics. You can transform a rectangle into a smiley face, smiley into triangle. You can reverse this process. You can transform a 2D origami into a 3D origami. It's quite interesting. You can have a flat object that totally transforms to become here a toroid that is hollow inside, so you can encapsulate something. And you can do the opposite. You can transform a 3D object into a 2D one, for example, to release something. You can do some kind of back and forth transformation, like from one shape you go to another shape and you come back to the initial shape. You can do multi-step transformation, even 2D, 3D, 2D. So you can play with the programmability. The most important thing is that we can transform objects by following this stability thermodynamic principles. And of course, then if you imagine you put some interesting object on it, like particles or proteins, then you can start to a very nice nanomachine, very nice things. We are also doing this, I will not comment too much, but we are doing this evolutive origami to discover optimized structure. So usually people design structure, but maybe if the system can select itself its optimized shape, then maybe we can discover like new origami with better properties. And for that, there is something very interesting in DNA, is its sequence. Okay? So usually, when you want to do an origami, you design the shape, you make the design, and you build your object. This is what we do. Most of people. But there is something, you can do the opposite. Imagine you find in the street an origami, right? We see dogs, but maybe you will see an origami. What is amazing is that if this origami has a very nice property, you can sequence and you know the composition, and because you know the composition, you can make it again. So you know, for example, people look in the ocean, they find a sponge, and in this sponge, they will find a strange molecule, and it happens this strange molecule is a very good anti-cancer drug. Okay, you know that this is how drug discovery is done. They are looking for molecules that exist in nature, that are very complicated, and then they will find them some properties. Oh, this molecule is great. You do mass spectroscopy, you do NMR, you know the structure, and then you are like, oh my god, how I will synthesize this object? It's very difficult. But here, if you sequence your object, because it has all the information, you can fabricate it immediately. So that's something very cool in this library, that you have the information that is inside. So then you can imagine that you have a target, you put a library of DNA, then you have a you have an optimized object that will assemble by itself, and then you capture, and then you sequence, and then you have discover a new object that could have very interesting properties. So a little bit like directed evolution, but on structure rather than on function. And you see that, so that's something also that starts to be very interesting when it's at the summarize. I told you already, I like very much this campus because there is a lot of plants. I like plants very much, and I like plants because they grow. And if you do conventional origami, you are limited. You are limited because you assemble and then it's finished. So, but if you are isothermal, maybe you can think of this assembly could continue and continue and continue. So maybe you can have assembly like 4. Okay, so we did that, and of course we don't do origami, because origami you have the scaffold, so you cannot go larger than the scaffold. So we did a very simple system, DNA nanotubes. So in the DNA nanotubes, you have just 5 oligonucleotides. I will draw because now it's drawn here, but maybe the scheme is not so easy. So you have 5 oligonucleotides. And they assemble like that. So you have one on the top, one on the bottom, one in the middle, and you have on the edge like that, like that. So you have 5 oligo, okay, they assemble like that. And what is special is that the sequence here, you have the complementary here. And the sequence here, you have the complementary sequence here. So this time, with 5 oligo, can self-assemble. Okay? Can self-assemble, and by self-assembly, they will make this large object. It should be a plane, it should be a flat assembly, but there is a little bit of curvature by design because of the double helix, and then it will close itself and it will make tubes. Okay, so it's just 5 oligos that make this pack, and self-assemble and make these tubes. And because it's isothermal, I can just wait. I don't have to do anything. And when I wait, these tubes are growing and growing and growing. And they grow so big that you can see by eye and by microscope. So you see these tubes that are growing and it becomes very, very long and very long. Look at this guy, like they can be like 50 micrometer up to 100 micrometer, these tubes. So they become very long. But at the same time, you have this very precise nanoscale programmability. So you see that if you do, we did cryomicroscopy. So you have this very giant nanotubes, and you do cryo-electron microscopy. I don't know if you see from the back, but I can see here very clearly the inner structure. I don't know if you see, but you see all the individual strands, and they are perfectly well positioned. So you see that this motif here is actually what you have here, and what you see is this part. Okay, and it's a bit like this shape. So you have this very nice control at the nanoscale level that can grow to very large dimension. And you can also play with self-organization. So if you put it in the synthetic cells, at the beginning you have tubes, these tubes grow, but at some point they will be confined. They cannot grow more than the size of the synthetic cells. So instead of growing, they will self-assemble, and they will form this kind of interesting organization, like you have some rings, some cross points, so they bundle and they do this dynamic organization that looks like a skeleton. This is something we can see. So we have kind of also objects that grow because we are fixed at some assembly. We can also think whether we could do an assembly that is not based on base pairing, but on something else. In fact, in nature, DNA is assembled not by hydrogen bonds, but by electrostatics. DNA is as I told you, it's a very charged molecule, largest charge density in the world, living world, and it has to be packed in very small dimensions, a few micrometers for cells. And the DNA, you have about 2 meters long of DNA to package in a few micrometers in the cells. And in a virus, you have the size that is about 100 nanometers. So it's very small, the virus space. It has to package all the genome. So DNA has to be very packaged. And this is done by electrostatics in both cases. And then, if you look just at the situation of viruses, if you just take double-stranded DNA in culture, if you add multivalent ions, DNA is spontaneously condensed at some point. We demonstrated a long time ago that is the first order phase transition with coexistence. And then this object, DNA is a charge, so when it condenses, you should make a sphere to be as small as possible, but because of its persistence length, because of its local rigidity, the optimal shape that balance the surface area, so you want to make it as small as possible, and the bending energy is actually a toroid of this mathematical shape. This is what the DNA is doing, so it's spontaneously form a toroid, and the radius of this toroid is the persistence length of DNA, so it's 50 nanometers. And we demonstrated that this is controlled by electrostatics, and whatever the conditions, this phase transition will always occur at neutralization. That is about 90% of DNA charge neutralized by DNA. So we are thinking like, this happens in nature. This is the first observation of a DNA toroid, and this is cryo-electron. You see how well DNA is organized if you do that. So this is very just spontaneous organization when you add multivalent salt, and if you do cross-section by cryo-EM, you see this very nice organization of DNA. So it's what DNA can self-assemble itself very precisely also. So we thought maybe we could do that. Maybe we could just take our giant nanotubes and just add spermine and see if we can make toroids. And yes, we can make toroids, you see. It's DNA to spontaneously form toroids. And the size is larger than the one of DNA because of the persistence length of nanotubes. These tubes have the persistence length of a few micrometers, and then you are going that are much larger. So this is what happens if I am at the dilute regime. I have some spermine self-assembled from these tubes. I know if I go to higher concentration of DNA, so I have my nanotube. And you see that when I add the spermine, I have this network, three-dimensional network. So this assembly of DNA can expand through the solution to form this network. So now you have this interesting phase diagram. Like when you're in dilute regime, you go from nanotubes to these toroids, and when you are in a concentrated regime, semi-dilute regime, you have again this two regime of nanotubes and rings, but in between, you have the formation of these very interesting networks. And you see that if you look at cryo-EM, when you add spermine, you have this very dense assembly of DNA that is actually very similar to what you find in nature. And because it's electrostatics, you can make it reversible, you can make the results with ions, so you can assemble, disassemble with ions, or you can use light to control this assembly whatsoever. Not so important. So it's also possible to do isothermal assembly by using principles other than base pairing. And I will finish the talk by just showing something a bit really alive. Okay, it will be more than lifelike, it will be alive. Because now we have possibility to build origami in very mild condition because it's isothermal. So far it was room temperature, so nothing harsh for cells. Until now, people use a lot of origami with cells for many kinds of applications, vaccines development, virus capture, so many things. But they always make the origami before they apply to cells. And we wanted to know, can we assemble origami directly in the presence of living cells? So try to remember, I said what we did to have origami assembly is to use monovalent ions and monovalent ion only, 100, 150 millimolar. But living cells, they need a bit of magnesium, they need a bit of calcium. But we know that if you put too much, then we'll have a lot of kinetic traps and the system will never self-assemble. So we look whether there are some conditions of cell culture where you have the good concentration of salt, of monovalent salt, and just enough multivalent salt for cell maintenance. And in fact, if you look at cell culture media, most of the cell culture media perfectly respect this condition. So we didn't do anything. We just took commercially available cell culture media. Okay, we don't do anything. We take cell culture media, we put the molecular program inside, 37 degrees, and that's it. And we look at what we get. And we get DNA origami. Without doing anything, without heating, without cooling, without nothing, just spontaneous assembly. And what is amazing, to me at least, because I was sure it would work, because there was no reason it should not work because of the electrostatics, but I could not expect it would work so fast. So you see that you have origami that form in 5 minutes. You have to imagine that everywhere in the world, people do thermal annealing and so on and so forth. Usually people prepare in the evening and they will take their origami on the following day. Sometimes it takes 2-3 days, especially if the origami are complicated. I know they form in just 5 minutes. They form in 5 minutes and you don't have anything to do, you just put in cell culture medium. So you see that biology is quite well done. Take cell culture medium and you can have this super fast self-assembly. And this can work directly with the presence of cells. In the cells, origami are happy, cells are happy. Then the origami form in just a few minutes and they are stable. And you can do even in the presence of brains. We grow not only origami in the lab, we are also growing brains. It's called brain on a chip. So you can have brain growing on a petri dish. And we take this brain growing on a petri dish, and we can assemble in situ these DNA origamis. So now you see that we have this method of isothermal assembly that is really more close to lifelike conditions, fixed temperature, reconfigurability. You have even origami that can transform. And you have all this possibility to have dynamic scaffolds, discovery of structures, growth, and self-assembly with the presence of cells. And I will skip the last part because we are already a bit late. I will just show you a movie. We were able to make crystals that are photoactive and that can grow with light. So look at this movie. We have a crystal. It's an assembly of a photosensitive DNA intercalator and nucleotides, so a brick of DNA if you want. You see, I put light. This is real time. I put light, my crystal is growing, I remove light, it stops. Okay, so I will not want to give too much detail, but I found this movie quite interesting. Anyway, so this is where we are in these 2 lectures, yesterday and today. I show you that it's possible to build fully synthetic materials that can have programmability, thanks to synthetic DNA, reconfigurability. We saw a lot of moving things yesterday. We saw origami that can transform today. So we have the origami. We have the evolution, the possibility to have this origami that transform and evolve. Response behavior. Yesterday we saw motion. And you see that when you control structure, you can have now some dynamic and adaptive DNA nanotechnology. And if you control functions, you can genetically encode soft matter, so bring a lot of function. And what I want you to remember is not what I'm doing, because it's not so important. What I'm doing is just my business. But what I found interesting is this idea of programming things using this DNA, as a tool to program. And you can program functions through self-free expression, and I think there's a lot of opportunity in this direction. And in the other way, you can program structure, and you have a lot of opportunity in technology. And as a soft matter scientist, as you are, it's 2 fields, DNA nanotechnology and synthetic biology, where there is very, very few soft matter people, very few. In synthetic biology, most of people come from either biology background and a bit of biophysicist, but it's people who just want to make equations. And in synthetic biology, it's people who just want to program complicated things, artificial genomes, gene circuits, all this kind of things. So no soft matter people in this field. And I think there are a lot of things to do, because self-assembly, membrane, you can make maybe models, systems, and you can use these properties to maybe make a smart gel, maybe to make some interesting polymer, maybe to make, as I showed you yesterday, some droplets that can have interesting behavior and so on and so forth. So there's a lot of things from the soft matter perspective in synthetic biology, and there are very few people. And on the nanotechnology, it's amazing that this field that is now 20 years old, most of people are come initially from computer science, and then came a lot of chemists, and a lot of, so chemists will do, for instance, sensing, will biosensing, or they will do catalyst, or they will do photonics, or physicists that they will assemble particles. But all these people, they are going to material science, but there is no soft matter people, very few. And you see that with my soft matter background, we were able to make really a breakthrough to change this way of self-assembling DNA. So I think you have also a lot of chance to make many kinds of breakthrough in this field. There's a lot to do from the soft matter perspective in DNA and technology, because we are using DNA, it's a soft matter object, and you are building using self-assembly, it's a soft matter principle. So there is also a lot of things to do in the understanding, or in the application. You can use this origami as a tool, just as a tool. You don't have to do research in origami. You can just take origami as a tool because you want to put particles. You are interested to, I don't know, you want to make cells, you know, or you want to make a Raman, and you want a hotspot between particles to make Raman spectroscopy, for example. So Raman is a great discovery in India. So, you know, you take two particles, and with my origami, I can decide at the distance I put these two particles, like 2.3 nanometer. And I can decide to put a single molecule wherever I want. I can even control the height of this molecule. And I can put a fluorophore, whatever molecule. So it's really a very powerful tool. So with that, we can stop, have a break. And I think the second session will start at 11.
Yeah, so maybe, yeah, 11, we decided 11.
As you decide, you are the boss.
Okay. Yeah, so thank you, Professor Bagel, for the wonderful talk. Now the floor is open for questions. If you have some questions.
I suggest, because this afternoon we will do this challenge, so we'll have time.
Yeah.
So because it's good that they have a break, you need a break. So let's have the break. I can have the question the beginning this afternoon.
Yeah, sure.
Okay.
Yeah.
Okay. So let's do that. Let's have a break.